let's do this. Okay, girls, I'm going to go over some of these questions, okay? Yeah, so one question, please, to start. So one question will be a monohybrid cross. So something like 1, 2, or 3. One question will be like a dihybrid cross, which is what we left off yesterday. And that's like question number 4 in your set. And then the third one is probability, which is something that has, well, really this is probability, but I'm going to show you some other examples with probability, okay? Like, for example, a question could be like, if a person has 8 kids, what are the odds that 5 will be boys? Okay? You can't really do that. I guess you could do it some way, but with the Punnett square, but it would be very difficult. I'll show you how to do that question later on, okay? So, let's look at this first one. We have a sepia is recessive to wild. Wild is red. So we're going to make red dominant because it says sepia is recessive, which means the other one has to be what? Dominant. So we're going to say capital R is red, and we'll make little r sepia. Sepia is brown eye color. Okay? They're all brownish eye color. So it says a sepia male mates with a heterozygous red female. So if a male is sepia, that means that the male must be little r, little r. The female is heterozygous, so she must be big R, little r. So if we do the Punnett square, we have, you look at the female can make 2 types of egg cells. One will have a big R, and the other one will have I shouldn't say will have, but may have 50% chance, right? The male can only produce little r's, so the sperm cell will only have a little r. Now remember, if the columns are repetitive, you don't really need to do it, but I'm gonna do it anyway. So this is an egg cell, this is a sperm cell, this would be a zygote, this zygote would be big R with a little r. This zygote over here would be a big R with a little r. These two would be little r with little r. You can see why I wouldn't need to do this column, because the probability is 2 out of 4 if I look at the whole Punnett square. But if I just looked at the first row, it would have been 1 out of 2, and those are exactly what? The same. So for the F1, you would expect 50% to be what? 50% of the flies to have red eyes, and 50% will have brown eyes. Okay? So if you look at the next one, but don't write this, I'm videotaping this. I can't wait for you guys to copy this. Okay? The next question, you have a pea plant is tall is dominant to recessive. So we have a purebred tall. Remember what that means? That means that they have 2 big T's, is crossed with a purebred short. Tall is dominant, short is recessive. So the F1, so these are the parents, parent generation. The F1, I don't need to do a Punnett square here, because this parent can only pass on a big T, and this parent can only pass on a little t. If you take the F1s, that's a different story, because the F1s are all going to be heterozygous. So this is a familiar, there's only so many patterns, right? So we kind of already know what this pattern is going to look like. When you get a heterozygote, we know they can pass on 2 different alleles, right? So it can either be a big T or a little t. The other parent is exactly the same, so big T or little t. So this pattern is a familiar, let's pick a different color here. This pattern is a familiar one. This is going to be big T, big T, big T, little t, big T, little t, little t, little t. So, how many will be tall in the next, or the F2 generation? This is your F2. Your parent generation gives you F1. The F1 gives you the F2. So this is the F2. So, yeah, 3 out of 4 are going to be tall because 3 out of 4, this one, and this one have at least one capital T, right? And 1/4 is going to be short. There's that 3-to-1 ratio that Mendel saw very often when he was looking at his studies with pea plants and stuff like that. In the next question, cystic fibrosis is a recessive condition. So what? Yep. Oh, okay. So that's for genotype ratio. Okay, genotype ratio is just the ratio of the genotype. So you would say how many are homozygous tall? 1. How many are heterozygous? 2. How many are homozygous recessive? 1. So the ratio is 1 to 2 to 1. Okay? Thank you, Pam. So for the next question, you have cystic fibrosis is an autosomal recessive condition. Now, we'll talk a little bit about why something's recessive and why is this recessive and why is that one dominant. So for cystic fibrosis, I'll explain this one. What happens is a cell has these things in its membrane, and sometimes these things are they act like, well, there's a lot of things in the cell membrane. Some of the things are transporters, doorways, okay? And what happens in cystic fibrosis is one of the doorways malfunctions. So this doorway actually allows a cell to remove chloride. So if a doorway is normal, the cell can get rid of salt, because chloride is an ion of salt. If the doorway malfunctions, what happens is well, it depends. Because you have 2 sets of DNA, if you're heterozygous, actually let's label this. Let's say this is normal, okay? And little n is the disease. So what happens if you have the disease? Well, you have 2 sets of DNA. On both sets of DNA, you have the gene for the transporter. If you remember, the name of the gene is CFTR. Okay? If you don't remember, not a big deal. This gene gives you the chloride ion transporter. That's the doorway to get rid of the chloride ion. If both copies are messed up, if there's a problem with both copies, and if you think of a copy as a recipe, anytime a cell goes to make the transporter, it malfunctions because the recipe there's something wrong with it. And both copies are bad, so all the CFTR transporters don't work. But if you're recessive, something interesting happens. You have 2 copies. Sorry, meant if you're heterozygous. If you're heterozygous, you have this scenario. One of the copies is being made from one of the recipes that is bad. So anytime that transporter is made, it doesn't work because that recipe is bad. And that would be the one coming from this allele, the little n. But because you have another copy, you're heterozygous, the other recipe is fine. So you end up making another transporter that actually works, where the ions can actually go through. So if you're heterozygous, you're fine because half the number of transporters is good enough for you not to have this disease. What happens in this disease is the cells accumulate salt. And what happens is, if you think of your airway, the cells accumulate salt. They take I don't know if if you remember osmosis, but what happens is, if you think of this as your airway, and these are the cells that line the airway, because they accumulate salt, they pull water into the cell. Well, if we have mucus that lines the airway and we take water out of the mucus, it becomes very, very thick and sticky, and it starts to impede breathing. So if someone has cystic fibrosis without medical attention, it could be fatal. Now, there are medications that you can help deal with this. So the reason why this is recessive is because for you to get the disease, you have to have 2 bad copies. Okay? 1 bad copy is not enough to give you the disease, because having 50% of normal transporters is good enough for you to avoid the disease. Okay? So, if you have 2 carriers, what are the chances that a child will be a carrier? Well, again, this is a cross between these 2 individuals. Now, we already know what that Punnett square pattern is going to look like, because is this any different than the one we just did? No, it's not, because you have a big N or a little n, you have a big N or a little n, so that's going to give us the exact same pattern as the one we just looked at. You'll have homozygous, homozygous dominant, you'll have hetero, and you'll have someone with disease. The odds of getting a carrier then is what? 1 here and 1 over here. There's 2 of them, right? Yeah, exactly. So make sure you read the question, because, yeah, it's a good point, right? Normally someone asks you for the disease. Here I ask you for the carrier. So the odds of getting a heterozygous individual is, you can see, 2 out of 4 or 50%. Okay? All right. Last question. Now this one's a dihybrid cross. So we have 2 types of characteristics, right? We have what? Color, and what's the other one? We have purple versus what? Versus white. Okay. And we have round versus wrinkled. Okay. Now, just so you know, because this doesn't say in the question, so I'm going to tell you this. Purple is dominant, white is recessive. So we're going to label this as big P, and this is little p. Round is dominant, and wrinkled is recessive. Now, 2 characteristics. Each characteristic is controlled by 1 gene. Each gene has 2 alleles, 2 versions. Yeah, there. In this case, you don't know. I know, because this is one of the experiments that Mendel looked at. You wouldn't know. You have to be told if it's dominant or recessive. Now, I could give you a hint. I could give you a hint. Say, like, for example, imagine I took a purebred purple and I mated it with a purebred white, and all of your offspring were purple. Then that's telling you purple is dominant. Okay? But if I don't give you a hint, you'll never know, unless it's something that we looked at. If it's something that we looked at, then I would expect you to know it. Something that we didn't look at, no, you need some information. Okay, so purebred purple that produces round seeds, what would the genotype be for that individual? Yeah, be big P, big R, and the other individual or plant would be white with wrinkled. That would be, white is recessive, so it has to be 2 little p's, and wrinkled is recessive, 2 little r's. Okay? I would recommend on a test or a quiz that when you write a big P, you write it very different than a small p. Do not write your big P like this and your small p like this. You know what's going to happen on a test when you're stressed out? You're going to think of the same letter. Okay? So, to get around it, if you want to even, you know, sometimes you have letters where the uppercase and lowercase don't look anything, like an L and a lowercase L. They don't look like each other. You can do that if you want. But if you have 2 letters that look like each other, really emphasize the difference in size, okay? Because otherwise you're going to make a big mistake. And you don't want to do that. Okay. So here are my parents. The F1 has to be what? I'm not doing the Punnett square for the F1, because there's only one possibility. It's going to be a big P with a small p.
Sorry, just your first set is wrong because you're using the F1 phenotype. So it would be purple with wrinkles and then yeah, it would be
Wow.
P, like big P, little p, and then R, big R, little r.
What?
For here, because look, if you cross the purple that produce round with white that produce wrinkles, determine the F1.
Yeah, that's what we're doing right now. That's what no swearing, please. This is a Catholic school.
Okay, I thought you were doing the second part.
That's okay. All right. We take confused students in our classroom. So that was Vanessa Watson.
You know what?
So we can see that the only possibility for the genotype of color has to be big P, little p, right? For round versus wrinkled, you're going to get one big R from this individual, and you're going to get one little r from this individual. So the genotype for F1 has to be a big P, little p with a big R, little r. Has to be. When we look at then the next generation, so we do this dihybrid cross. Well, okay, how many genotypes are possible? This is what we were talking about yesterday. Both these codes are the same, right? They're both big P, little p, big R, little r, right? It's the same. So if you think about the possibilities, one possibility we said yesterday is a sex cell could have the big P paired up with the big R. So that's one possibility, big P with big R. Another possibility, big P with the small r. A third will be the little p with the big R. Yes, it is. I was just seeing if you're paying attention. Third possibility is a little p with a big R. And then the fourth possibility is little p with little r. So you can see, sorry, little r. Yeah, little r. So you can see there's four possibilities in this case. Okay? So big P with big R, big P with little r, little p with big R, little p with little r. Yep. No, that was in the first generation, the parents. Parents produce the F1. The parents are purebred. So you can see purebred means that the alleles are the same. So purple for one parent and white for the other.
No, it says if an F1 to the previous cross is related to the purebred white,
oh, yeah, you're right. Okay. So, yes. Yeah, you're right. I thought it was two F1s being mated. So we're going to mate, okay, so yeah, moral of the story, read the question. So we're going to mate the purebred, sorry, we're going to make the F1. So one of the F1s is going to be mated with a purebred white that produces wrinkled. So that would be little p, little r. I apologize. I thought it was the two F1s being mated. I should have read the question. The F1 is purple wrinkled. Purple round. Right? Because what do you see, Kiara? This is the F1 right here. So what do you see here? What letter? Well, and big P stands for? Okay, what letter do you see here? And that stands for round. So the F1 is going to be purple and round. We're going to mate it with a purebred white that produces wrinkled. So we're going to mate the F1 with white, which is little p, and wrinkle, which is little r. We just figured out the F1 can produce 4 types of sex cells, right? The big P with the big R, the big P with the little r, the little p with the big R, little p with the little r. So if you do your Punnett square, how many rows or columns, we'll look at the column. We'll do the F1 as the columns. Sorry. We'll do the F1s up here. So the F1 will gives you 4 possibilities. Big P with big R, big P with little p with big R, little p with little r. If you look at the white wrinkled, how many possibilities are there? Well, there's only one, because you're only going to get a little p with a little r. Look, there's only little p and little r's to be donated. So there's no, you don't need to do a 4x4 grid here. A 4x1 is fine. So then, what do we get? We got big P with a little p. Just put the letters together. Big R with a little r. Big P with a little p, little r, 2 little p's. That sounded kind of funny. A big R with a little r, 2 little p's, and 2 little r's. So what's this? What is big P? At least one big P and one big R. That is what? That would be purple and round. Okay. This would be purple and wrinkled. This is 2 little p's, white and round. And this has to be white, 2 little r's, and wrinkled. So the ratio here is 1 to 1 to 1 to 1. 25% chance for each one. Okay? And then we'll continue with this tomorrow. Look at that, perfect timing. The end.